Thank you for introducing me, and welcome all of you for this seminar session. Before we start talking about AMR, I would like to start with something very simple, just a simple challenge. Look at your last meal you had this morning or yesterday. Which animal sourced food was on your plate? Is it maybe milk in your coffee, cheese, yogurt, maybe chicken, egg, beef, or any combination of this? Now imagine this. What if the effectiveness of the medicines we rely on to treat our illnesses has been slowly modified by what happens during the production of this animal origin food? So this presentation is entirely about that hidden connection. We will see how resistance genes circulate among human, animal, and the environment interface, with a particular focus how the animal sourced foods interplay in this exchange. So over the last 20 years, the world has faced numerous emerging and re-emerging health threats. Many of them have been eliminated. Some of these threats has been reduced in their intensity. However, AMR remains a persistent and unresolved paradox. Despite the ongoing global efforts, it continues to rank among the leading public health threats worldwide. So this highlights the need for sustained attention, coordinated action, and continued dialogue in this silent pandemic. So AMR is a borderless and escalating global health crisis, and WHO has recognized it among the top public health threats of the 21st century. And it is driven by multiple interconnected and cross-sectional factors. But in most of the sense, healthcare settings and pharmaceutical effluents are often regarded as the epicenter of AMR. However, growing evidences indicate the gut of food-producing animals is a major reservoir and application site for the resistance gene development and dissemination. So this seminar explores the role of livestock in the emergence and dissemination of this silent pandemic. It will integrate established evidences, and I will present findings from our own research works, just to offer field-based insights, of course, from low-income settings. So the AMR has numerous global impacts. Is causing millions of deaths, and it is also responsible for longer hospital stays, which highlights the difficulty of managing resistant infections. This can incur additional healthcare costs. So these additional healthcare costs, along the loss of productivity, is putting a high economic burden on individuals and the nations. Is also responsible for poor health outcomes, which at all reduces the quality of life. So AMR has a huge burden both on the public health and the economy. The existing trend shows the deaths which are directly due to AMR rises 1 million per year, and deaths which are associated with AMR are about 5 million per year. Unless effective measures are implemented to prevent and contain this crisis, both of the burdens are estimated to rise over time. So this is the model-based estimate in the coming 25 years, from 2025 up to 2050. It shows more than 39 mortality will be occurred, which is directly due to AMR, and this figure excludes the diseases associated with AMR. So as of 2050, the annual diseases will be continually 10 million, out of whom about 2 million are directly associated, directly due to AMR, and the remaining 8 million diseases are associated with AMR basic infections. In addition to this public health burden, the World Bank has also estimated that AMR could result in $1 trillion USD additional healthcare costs by 2050, and up to 3.9 trillion USD GDP losses will happen as well by 2030. So this is the regional distribution of the estimated 10 million diseases as of 2050. The highest burden will be in Asia, followed by Africa. Europe, Latin, and North America will have more or less similar burden. So having this, the impact of AMR both on the public health and the economy, countries are reacting against this AMR very differently. Only 149 countries have developed AMR National Action Plan. Out of this, 66% of them are really implementing their National Action Plan. Of course, it is at different level. Some of them are at level C, which is they are starting the implementation. This will advance to level D, and level E indicates the full implementation of the National Action Plan, which is really implemented by 70 countries only. So this shows how the countries are advancing or progressing both in developing their AMR National Action Plan and how they are implementing it. In 5 years, from 2017 up to 2022, the development was from 41% to 77%, whereas the implementation is lagging far behind the plan, which indicates a real gap between policy development and the actual implementation. So what are the anthropogenic drivers of this AMR? Of course, AMR has been driven by many parameters, but the irrational drug use in animals, agriculture, and human are the top reasons that induces AMR. Once bacteria has got selective pressure due to this irrational drug use, they will develop resistance due to different mechanisms, particularly by genetic mutation, gene transfer, and physiological adaptations. This magnifies the mechanism of resistance differs across the bacterial strains. This is the method which is commonly employed by many bacteria. They reduce the drug uptake to the intracellular environment, and they can do this by modifying the membrane molecules, particularly the porine channels. And they also activate their intermembrane proteins. This activated intermembrane protein will further activate the drug efflux pump. So this activated drug efflux pump will remove the drugs from the intracellular of the bacteria to outside, which reduces the concentration of the bacteria in the intracellular environment. Some bacteria do also develop enzymes that degrades the antibiotics. Then degraded antibiotics cannot proceed with their real pharmacokinetic activity. They also modify their target site, so this blocks the successful binding of the drug to its actual site. And the target modification can also be facilitated by genetic mutation. For example, this group of drugs are indicated to disconfigure the DNA structure and further reduce the DNA replication, but the resistance bacteria do modify their genes, which are encoded to these enzymes. So these groups of drugs will fail to connect to successfully bind with their actual site. So once the bacteria has developed resistance, the resistance genes will not confine to a single bacteria. It will further further distributed to the bacterial population. It can be by horizontal gene transfer or vertical gene transfer. Horizontally, the resistant gene can be transferred from the donor bacteria to the recipient bacteria. In one of these three mechanisms, it can be by transduction, which is facilitated by the bacteriophages. Bacteriophages can carry the resistance gene from the donor to the recipient when they change their host cells, bacterial host cells. It can also be facilitated when the recipient and the donor bacteria come close together, and the donor bacteria can transfer the resistance genes to the recipient via the sex ply. Further, this recipient bacteria can also pick a resistance gene from the environment, which is released by dead or live bacteria. Vertically, obviously, resistance genes will transfer from the parent cell to the daughter cells during the replication of the bacteria. So this shows the event when Paul Ehrlich has developed the first chemotherapy in 1907, the time when this chemotherapy has been clinically officially tried in a medical setup. And this shows the subsequent years when different antibiotics has been discovered and developed. And this shows the biological counteracts developed by the bacteria. When the striking news comes in 1940, where the penicillin enzyme has been discovered from a resistance bacteria, this penicillinase enzyme will activate the action of penicillins, which is the first antibiotic discovered in the medical history. So the first resistance has been observed against the first drug, which is penicillinase against penicillin. After this observation, drug resistance follows in a very short years after the discovery and development of new drugs. So this scenario has discouraged a lot of pharmaceutical industries, so they decline their development of new drugs. This can be manifested, for example, in the last 20 years, only 5 novel drug classes has been discovered, which is very shocking. And even some of the pharmaceutical industries has switched their role to develop drugs which are used to treat the non-transmissible infections, which is the so-called lifestyle diseases, because it's more profitable and there is no risk of drug resistance in this area. So what will be the particular role of livestock in this transmission? Of course, in the development and transmission, they do have a great role both in drug development and in resistance development, and it is a transmission of these resistance genes. So the gut microbiome of food-producing animals is a very stable ecosystem, but this can get disturbed by resistance genes. Once a resistance gene has been introduced to this gut microbiome of the food-producing animals, the gut will be changed into a melting pot, and this can further transfer the resistance genes horizontally to different organisms. There are different methods. So drug development can, resistance genes can get developed within these food-producing animals at farm level through these different mechanisms. But the first and the most will be the intensive and irrational use of drugs during the livestock farming. This can be used by using drugs as growth promotion, which is most commonly used. These drugs as growth promotion can be used directly by injecting these drugs to the fattening animals, or it can also be used with food if the intention is to deliver these growth promoter drugs in large extent. Further, drugs can also be used as a prophylactic treatment in livestock production. This is where drugs are used to healthy livestock in some risky areas or some risk periods. If there is some fear of outbreak dissemination from neighboring zones, or due to the epidemiological alarming of some fears of emerging or re-emerging diseases, which is playing a great role for the development of the resistance, because most of the time the drugs are used in their lower doses. Drugs can also be used as metaphylactic treatment. Drugs will be entirely given to the whole herd when a single or few animals have shown some clinical signs. So the paraprofessionals or farmers will prefer to treat the entire herd, even those animals which do not show clinical signs. And in this poultry and swine farming, drugs can also be used via the water systems. On top of this, the poor disease prevention and control mechanisms can also play a great role in the resistance development, because in this case, if we break the prevention and control systems, the incidence of disease in trans will be increased. That will push us to further use intensive antimicrobial drugs. Even though about 74% of the countries have banned the use of drugs as growth promoter in 2015, at this time, 2/3 of the antibiotics used globally are consumed in the livestock farming, and most of them are used for non-therapeutic purposes, either to promote fattening, or they can be used as a form of prophylactic treatment or metaphylactic treatment. And most of these applications are used irrationally. This further highlights the difficulty of implementing what really we are inspiring to do. So this systemic review and meta-analysis of the global antibiotic use in livestock for 10 years indicates that the global consumption of antibiotics has been increased by 67% in 10 years, from 2000 and up to 2010. And based on this existing situation, the consumption in livestock will further increase by 53% in 2030, which further complicates the development and dissemination of the resistance genes. This is one of our research findings that has been conducted in fattening farms, which supplies sheep, goat, and cattle to the abattoir for slaughter. And the intention was whether these farms were using drugs for growth promotion or not, or how they were implementing these drugs. We also further the study to isolate Staphylococcus aureus, one of the potent zoonotic bacteria, and we have evaluated these Staphylococcus aureus strains against the different drugs which are used in that district, both to treat livestock and human. And the finding shows that the fattening farms were using drugs for growth promotion, and the Staphylococcus aureus we isolated from the same species tract has also developed resistance against the commonly used drugs, which can be possibly linked to the irrational use of these drugs on the farm level. Of course, it needs further study to confirm the actual causality, because there are also other confounding factors on the development of this resistance. But the worst scenario, because this Staphylococcus aureus, which is zoonotic and resistance has been isolated from the meat, it can further be a source of infection for the consumers. And the bacteria is also isolated from the abattoir environment, and it can also has the potential to contaminate the environment via the untreated abattoir effluents. The antimicrobial resistance, the effect of antimicrobial resistance is very more in pastoral areas, where the access to health service is very limited, and the livelihood of the pastoralists is entirely dependent on the livestock and their products. They are very close to their livestock. for their survival. So our assessment in this setup in one of the pastoral areas in Western Ethiopia shows there was high level of drug mismanagement. Farmers were treating their animals by themselves, and the community animal health workers, the cows, who are not really trained professionals, were carrying out the significant part of the treatment in livestock and sometimes in themselves. And they were, both the pastoralists and the cows were extensively using drugs as a form of prophylactic and metaphylactic treatments. So, because the pastoralists heavily depend on their livestock, we further the study to look the microbial safety of their food. And milk is the major source of food in the pastoral areas of Ethiopia. And the major dairy animals in this pastoral setting are camel, followed by goat and cow. So we have isolated different bacterial species from these major dairy animals in that pastoral setting. We have also evaluated the microbial and antimicrobial profile of these isolators against the drugs which are commonly used in that pastoral setting, both to treat animals and humans. And this heat map shows the antimicrobial profile of these isolators. The color intensity shows the level of resistance, where red indicates the high level of resistance, orange is the partial resistance, and yellow indicates the low resistance. So almost all of the isolators have been found resistant to most of the drugs. But with this in mind, milk is still the sole food source of these pastoralists, and in the worst scenario, they consume the milk in its raw state due to some cultural and religious beliefs. So we can imagine that this can further complicate the issue of resistance, because some of the bacteria are zoonotic. They can induce infection on the consumers. On top of this infection, they also carry a resistance gene. This is how really the milk, which is the major food of these pastoralists, is being produced, marketed, and consumed. Is in a very rough hygienic status. But still, they prefer to consume it in its raw state, and most of the time immediately on the milking shed. So what will happen when we use drugs in a very irrational way? This shows how the resistance is actually developed inside this, particularly in livestock. This shows the concentration of drugs, and the time exposure in hours are expressed in the x-axis. The curved line shows the level of drugs has been raised after dosing. It will reach the peak and decline afterwards. So the peak indicates the maximum concentration. So when the maximum concentration of the drugs is below the minimum inhibitory concentration, this will have no effect on both the susceptible and resistance genes. When the concentration of the drugs is between minimum inhibitory concentration and mutant prevention concentration, there will be high level of selective pressure. So the resistant, the susceptible ones will be killed by this amount of concentration, whereas the resistant ones will stay and they keep multiplying. And this mutant selection window is a fertile ground, or the training ground for the bacteria to get resistant. When the concentration comes above the mutant prevention concentration, there will be an optimal killing, and both the resistance and susceptible bacteria will be completely died. But when drugs are used irrationally, the concentration of these drugs will be, in most of the time, under this window, the mutant selection window, or sometimes less than the minimum inhibitory concentration. And this strictly facilitates the development of the drug resistance within the bacterial population. So the resistance genes will be communicated among livestock, human, and the environment. The animals will distribute the resistance genes to the humans through their animal origin foods and to the environment via the contaminated manure. And humans can transfer back these resistance genes to livestock and the environment will give back what has received from the livestock and the humans through the contaminated food and water. And this is the particular focus of the animal sourced food. In the livestock rearing, in the rearing of these food-producing animals, drugs are used irrationally, either to promote their growth in short time or to in a form of prophylactic and metaphylactic treatment. These are grounds for the resistance development. So these genes, these bacterial genes who develop resistance due to this selective pressure will be further communicated to the food of animal origin during the production, particularly during slaughtering and milking. So this contaminated food will be a source of transmission of this resistance gene to the consumer. In most of the time, if they are, the risk of transmission will be worse when this food are consumed in their raw or partially treated way. Most of the time, we do share the same antibiotics with livestock. So when these same antibiotics are given to livestock, if they are given irrationally, the drug resistance will be developed under these food-producing animals. What has been developed here will be communicated to us through these animal-sourced foods. Then we could not be effectively healed in later infections with the same drug, because we have already acquired resistance gene which is particularly resistant to these drugs. So the resistance genes can be shared, antimicrobial resistance genes can be shared between us and our food-producing animals. For example, this result shows some of the antimicrobial resistance genes were shared between human and swine, some of them between human and chicken, and some of them in between these food-producing animals, and about 183 of the resistance genes were found in the gut of human, swine, and chicken. I'm sorry to display this slide, which is in your peak lunch time. But this is the traditional meat consumption in Ethiopia. It's a common practice to consume meat in its raw state or in partially cooked manner, and this is also practiced by different visitors. But this can contribute a source of infection if there are zoonotic bacteria and a source of resistance gene transmission to the consumers, unless these food are produced under a very strict hygienic situation, which is not easy in every setting, especially in the low setting countries. So to see the microbial quality of the meat which is consumed in this way, we have evaluated the microbial safety analysis. We collected the raw meat which is ready to eat from the table of consumers, and we have isolated different bacteria, but our particular focus was on these 3 bacterial species because they are zoonotic. They can induce infection. On top of that, they were found resistant to more than half of the tested drugs. So it will be a double burden. They are zoonotic, they can cause infection, and they can also carry and transfer the resistance genes to the consumers as well. So bacteria are aggressively striking against the antibiotics. Just they are surviving, they are fighting for their survival. They are calling their susceptible colleagues to join them, and they have realized that they do have two options. Either they have to evolve the action of the existing drugs, or they will die. But for us, we do have only one viable option, that is only to win the race. And this can be done entirely, at least by stopping weaponizing these microorganisms. The intensive and irrational drug use is enabling this bacteria to evolve and get resistant. So we have to stop this intensive and irrational drug use. And we can easily manage it by developing and implementing a very robust antimicrobial stewardship. This can be further grounded by strict biosecurity measures. So by doing this, by implementing plausible antimicrobial stewardship program supported by biosecurity, we can stop the resistance at the farm gate, and we can let the farm healthy, and healthy farm will be positively contribute to the health of the consumers and the environment. So WHO and the World Organization for Animal Health have outlined key priorities to reduce the impact of AMR in food producing animals. And this is mostly on the prevention of infection to these animals. And there is also a need to establish and implement the antimicrobial stewardship program, because this can optimize and reduce the antibiotic use. The stakeholders has to be aware and engaged, and there has to be a routine and strengthened surveillance system for antimicrobial use. And the food production has to be safe across the value chain. And we have to look for alternative treatments. This can be probiotic based or feed therapy. So the intensive drug use is facilitating the drug resistance development. So by reducing the demand for drug, we can positively contribute on the antimicrobial resistance development. And this can be done by different activities that focuses on the prevention of infection and behavioral change. And this shows the implication of working on strategies to reduce demand. This shows the age-specific deaths in human, in different age species, for 30 years, and the death was directly due to AMR or associated with AMR. And it has two extremities. Over the last 30 years, from 1990 to 2021, the mortality rate has been declined by more than 50% in the younger groups, particularly in those aged 5 years and below. On the contrary, the mortality has been rising in the older age groups. It was about 80% for those who are aged 70 and above. And the main reason what makes this reduction in these children was evaluated that the massively practiced vaccination for children under 5 years across the globe, because this is a global data, has positively contributed to the need, the occurrence of infection and the need for frequent antimicrobial use. And this was also further grounded by using the strong wash system, the water sanitation and hygiene. So this demand reduction for antibiotics can be also exactly conducted in the livestock environment as well. And this can be done by working on improved livestock production practices. And we have to implement biosecurity measures, which is tailored to different farming systems, because the biosecurity is tailored, it differs from farm to farm. So we have to adopt that tailored biosecurity measures, and this has to be supported by strong vaccination programs. So as a matter of translating the evidence into practice, we have done one interventional study on dairy farms located in the northern part of Ethiopia. It was a longitudinal experiment, and that was conducted on 22 dairy farms. 11 of them were included on the intervention, and the remaining half were put on the control. And the intervention took us 6 months, and it was mainly, it was a participatory approach, and the focus was to enhance farm management, drive to work to modulate their behavior on the drug use, and to promote the prudent antimicrobial use as a matter of reducing the AMR development and dissemination. Data were collected at the beginning, before the intervention, mid of the intervention, and at the end of the intervention. And here shows the impact of the intervention on the farms. Before the intervention, both the intervention group and control group had similar perception and understanding on AMR and antimicrobial use. After the intervention, the data shows both, particularly the first cycle of the intervention, shows that there has been the mean score of the perception and understanding of the farmers, the dairy farmers who were included in the intervention, has been significantly increased compared to the control ones. And these were the top dairy diseases in that district for that particular period, because some of them are seasonal. The occurrence of some of the diseases is seasonal, and some of them will endemically exist in the farm. So we selected these top dairy diseases, and we have evaluated the incidence rate of these diseases on those farmers who were included in the intervention and the control groups. So the incidence rate has been significantly reduced on the farms of those who were included in the intervention, except for lameness. And this is because mostly it was not. Of course, lameness can be occur both due to infectious and non-infectious causes, but in these dairy farmers, most of the time lameness was happening due to some mechanical issues. So finally, the demand for the antibiotics, which is measured by treatments for 100 cow days, has been reduced by the farmers who were included in the intervention. When we compared this demand to those for the control, they just keep using drugs for growth promotion. They were using drugs as a prophylactic treatment and for metaphylactic treatment as well. So once to conclude, AMR is a transboundary and evolving global health crisis, and the food production chain serves as a primary evolutionary corridor in which sublethal antibiotic concentrations create a selective landscape for the emergence and dissemination of resistance genes. In this context, animal-sourced foods are not passive products, but part of a dynamic system that actively modulates the human clinical resistance profile. Therefore, addressing AMR in human is not possible without ensuring the microbial safety and responsible antimicrobial use practices within the animal food production systems. By reducing the unnecessary selective pressure in the livestock environment, we can effectively slow the evolutionary trajectory of the resistance lineages, providing a biological shield for the consumers. Finally, effective mitigation of this multifaceted challenge necessitates both sector-specific and well-coordinated actions across human, animal, and environmental sectors, which is firmly grounded in the One Health approach framework. Thank you very much for your attention.